Hello everyone, welcome back. In last lecture we have discussed about transcription in eukaryotes. Today we will discuss one important topic in molecular biology, that is translation process in prokaryotes and eukaryotes. As we know that transcription and RNA processing are followed by translation, which is formation of proteins as directed by mRNA template. And as we know that proteins are the active players in most of the cellular processes, and protein synthesis is thus the final stage of gene expression. However, gene expression is not controlled only at the level of transcription, but also at the level of translation. So this is the important step in gene regulation process. और जो proteins बनती हैं as a result of translation, they will undergo some modifications. After that, they will be activated, or after their activation, they are sorted to the particular place where they are required. So this is the brief information regarding translation. In next slide, we have one animation that will give you an idea, brief idea about translation process. So let's start.
Protein synthesis, translation. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, mRNAs move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm is an enzyme, amino acyl tRNA synthetase. The enzyme macromolecule has two binding sites. One site recognizes the amino acid, methionine. This is followed by binding of the ATP molecule and release of pyrophosphate, resulting in activation of amino acid. Finally, tRNA and the activated amino acid bind together. This amino-acylated tRNA is known as met-tRNA and is released from the enzyme. This marks the commencement of the first stage of protein synthesis, the initiation stage. During the initiation stage, a small subunit of ribosome binds to the mRNA strand. The mRNA strand is made up of codons, which are sequences of 3 bases. Then the ribosome subunit moves along the mRNA in 5' to 3' direction until it recognizes the AUG codon or the initiation codon. At this point, met-tRNA, possessing the anticodon UAC, pairs up with the AUG codon of the mRNA. Then a large subunit of ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the acceptor site or the A site, the peptidyl site or the P site, the exit site or the E site. This whole unit forms the initiation complex. This is followed by the elongation stage. During this stage, another tRNA carrying molecule of an amino acid approaches the mRNA ribosome complex and fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. As a result, met-tRNA becomes deacylated. The ribosome then advances a distance of 1 codon, and the deacylated tRNA shifts to the E site from where it dissociates. Meanwhile, another tRNA carrying amino acid molecule attaches to the A site. This is followed by the binding of the amino acid molecules. Repetition of this process leads to the formation of an amino acid chain. This event is called elongation. Finally, when the UAG codon or the stop codon reaches the A site, elongation is terminated. Termination is the last stage of protein synthesis. The chain of amino acid molecules is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation. Then the tRNA detaches from the mRNA. Ribosome detaches and dissociates into its small and large subunits. Summary. Protein synthesis shows that the first stage involves the binding of the met tRNA to mRNA and the small subunit of the ribosome. The larger subunit of ribosome then combines with the small subunit. The second stage is the elongation stage. In this stage, the incoming amino acid tRNA fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. The process is repeated until a chain of amino acid molecules is formed. The last stage of protein synthesis is the termination stage. When the ribosome reaches the stop codon UAG, elongation stops and the newly formed amino acid chain, which is the protein macromolecule, detaches from the ribosome. Subsequently, ribosomal subunits, along with the tRNA, dissociate from the mRNA.
So what is translation? As we know that protein synthesis is accomplished through the process which is known as translation from the mRNA. So formation of polypeptide from this mRNA is known as translation. It is the formation of protein from mRNA, which is this mRNA is formed from DNA by the process of transcription that we have already discussed in last lecture. So central dogma biology, as we know, it is the formation of mRNA from DNA by the process of transcription and proteins from mRNA by the process of translation. So this process, this unidirectional flow of energy from DNA to protein through mRNA is known as central dogma biology. It is a unidirectional process. mRNA along with tRNA and ribosomes work together to produce protein. So इसमें तीन basic requirements हैं इसकी. एक तो mRNA, then tRNA which act as the adapter molecule, and the ribosomes which are basically the protein factories of the cell. These three together will produce proteins. Then site of protein synthesis. So translation takes place in cytoplasm, and the machinery which is used for this protein synthesis are the ribosomes. Next, what are the various requirements for translation process? Firstly, it requires activation of amino acids. For this, we have 20 amino acids which are known as magic 20 of life, and for that we require 20 amino acyl tRNA synthetase. These are the enzymes which helps in activation of or charging of tRNA, and we require 32 or more tRNAs. In addition to this, we require ATP and magnesium ion. So these are basic requirements for the synthesis of proteins. Then it involves 3 steps, that is initiation. For initiation, we require mRNA. The cell requires mRNA, then it requires a methionylated tRNA, that is 1N4-mile methionyl tRNA. Then initiation codons are required, and it requires ribosomal subunit. In case of prokaryotes, it is 30S and 50S subunits, which are the ribosomal subunits. Then initiation factors are required, that is IF2 or IF3. Then GTP, that is act as a source of energy, and magnesium ion. So these are the basic requirements which are required for the process of initiation, to start the process of translation. The second step in translation is elongation, जिसमें polypeptide chain elongate होती है. For this it requires the complete 7ts ribosome, जो कि एक कॉम्प्लेक्स बनाता है इनिशिएशन फैक्टर के साथ, जिसे बोलते हैं इनिशिएशन कॉम्प्लेक्स. और इसमें चाहिए अमाइनो एसाइल tRNA which are specified by codons और mRNA. जो mRNA के ऊपर codons हैं वो डिसाइड करते हैं formation of this amino acyl tRNA. Then elongation complex are required, Along with this, it requires GTP, which acts as a source of energy. Then the last step in translation process is termination, which means stoppage of protein formation or peptide formation. So for this event, termination codons are required, और जो termination codons होते हैं वो mRNA के ऊपर present होंगे. And along with this, we require release factor. The cell requires release factor for termination process. So there are various release factor which are present. Then it requires some post-translational changes which involve specifying enzymes, cofactors, and other components for this. So first requirement, that is ribosome, which are known as protein factories. We have discussed about ribosome in the previous lecture. These are large and complex molecular machines that serve as primary site for biological protein synthesis. So that's why these are known as protein factories of the cell. Made up of 65% rRNA, that is ribosomal RNA, and 38% of protein. So this is the basic chemical composition of ribosomes. There are two major components of ribosomes, the smaller ribosomal subunit and the larger ribosomal subunit. And for this dissociation of these units or association of these two units require the important ion, that is magnesium ion. So magnesium ion plays an important role in its dissociation and association of these ribosomal subunits. Two irregularly shaped ribosomes fit together, forming a cleft in which mRNA passes. It has 3 tRNA binding sites. जो ribosome है उसके ऊपर 3 tRNA binding sites होती हैं, which is acceptor site, abbreviated as A, then peptidyl tRNA site, abbreviated as B, and exit for deacetylated tRNA, abbreviated as E site. So these 3 sites are present on the larger subunit of ribosomes. It has different sedimentation coefficient in prokaryotes and eukaryotes that we have already discussed in the previous lecture. 70s ribosome is present in case of prokaryote, which is made up of smaller subunit 30s and 50s larger subunit. Then 80s ribosome is present in eukaryotes, which consists of 2 subunits. Smaller one is 40s subunit and 60s larger subunit. Then comes the tRNA, which is transfer RNA. We have already discussed about this RNA, that is tRNA. It serves as an adapter molecule. Adapter की तरह act करता है in translating language of nucleic acids to protein. So it acts as an adapter. And it is composed of 73 to 93 nucleotide residues. ये इसकी chemical composition है, having a molecular weight of 24,000 to 31,000. Minimum there are 32 tRNAs which are required to recognize amino acids with the help of codons. So जो codons हैं उनको read करके ये special amino acids को लाते हैं from the cytoplasmic environment. For this it requires minimum 32 tRNAs. And if we talk about its structure, it consists of various arms. The first arm is amino acid acceptor arm, which carries specific amino acids at the 3' end of tRNA. So this arm acts as the amino acid acceptor arm. Then anticodon arm, which reads the codons of mRNA. जो mRNA के codons हैं उनको read करती है ये arm, that is known as anticodon arm. And then comes D arm, which contain an unusual base, that is dihydrouridine. So this is present in the D arm. It helps in recognition by ribosome. So ribosome इससे recognize करता है tRNA को. Then T-C arm. So this arm also contain one unusual base, that is ribothiamidine. This arm also plays a role in recognition by ribosomes. इस arm में भी जो unusual bases होते हैं वो help करते हैं ribosomes को to recognize this tRNA. Both D and T-C arm help in overall folding of tRNA and interact with large subunit of ribosome. So ribosome basically interact करता है with this tRNA with the help of these two arms, that is D arm or T-C arm. Next stage is in protein synthesis. So first stage is activation of amino acid. For this it requires amino acid tRNA synthetase, which are esterified by 20 amino acids to corresponding tRNA. And each enzyme is specific for one amino acid and one or more tRNA is required. Then two classes of this enzyme is based on substantial differences in primary and secondary structure and reaction mechanism. Class 1, which synthesizes these amino acids, which brings these amino acids, and class 2, which brings the mentioned amino acids. This enzyme has also proofreading mechanism, which ensure the fidelity or we can say accuracy of protein synthesis. जो ये enzyme है ये प्रूफ रीडिंग भी करता है, which shows or which ensures the fidelity, which means accuracy of protein synthesis. The first step in translation process is initiation. In this process, the smaller subunit, that is 30S ribosomal subunit, bind with two initiation factors, which are IF1 and IF3. Then, this smaller subunit binds at specific position on mRNA, which are known as Shine-Dalgarno sequences. So, this mRNA binds to 30S subunit, which is guided by some special sequences which are known as Shine-Dalgarno sequences. और ये जो sequences होते हैं ये present होते हैं लगभग 7 to 8 nucleotide upstream from the initiation codon. So located about 7 nucleotides upstream from the initiation codon. जो initiation codon है उससे लगभग 7 और 8 nucleotide base sequences ऊपर present होते हैं ये toward upstream direction, and these special sequences are known as Shine-Dalgarno sequences. This complex joined with another transcription factor जिसे कहा था IF2 and with the formyl methionine tRNA. So जो formylated methionine tRNA है वो भी इसके साथ bind करता है, and this anticodon joined with the initiation codon on mRNA. और जो mRNA का initiation codon है उसके response में जो tRNA है वो methionine tRNA को help करता है इस initiation complex में bind करने. This large complex combines with the largest subunit of ribosome, that is 50S subunit, and GTP, which provide energy by hydrolyzing itself. So this is a source of energy. So after the formation of this initiation complex, जो largest subunit है ribosome की वो bind करती है smaller subunit के साथ. Before this process, जो IF3 है वो यहाँ इस complex से release हो जाता है. So before binding of this largest subunit to this complex initiation complex, जो IF3 है वो यहाँ से release हो जाता है, and after that IF1 and IF2 departs from this complex, and after formation of this complete subunit जो IF1 है और IF2 है ये भी इस complex से निकल जाते हैं. A functional 70S ribosome called initiation complex, जो ये complete subunit बनता है 50 और 30 का, so which is the functional subunit, और इसे कहा जाता है initiation complex. The addition of this 50S subunit, ribosomal subunit, to the complex puts this methionine tRNA, that is formylated methionine tRNA, in the peptidyl site. जो P site है ribosome की larger subunit पे, वहाँ पे जो tRNA है which contains formylated methionine. In case of prokaryotes वो आ जाता है directly P site पे with the anticodon of tRNA which are aligned with AUG, that is initiation codon on the mRNA. जो mRNA है उसके ऊपर यहाँ पे initiation codon लगा होता है AUG, उसके response में जो tRNA है that brings formylated methionine at the P site directly, और इस तरह के tRNA को कहते हैं charged tRNA, and for this charging it requires energy that is provided by GTP. This formylated methionine is the only amino acyl tRNA that can enter the P site directly, और ये this is the only tRNA, formylated methionine tRNA, जो directly P site पे enter करता है. Otherwise the next one will enter through the A site. Without passing through the A site, ये A site में नहीं जाता है. This first one it directly comes to the P site. बाकी जो next के tRNAs होंगे charged tRNA, they will come to the A site first, and after that they will enter the P and then finally to the E site. So the initiator codon, that is AUG, is at P site, and the next codon of mRNA is in Q at the A site. और जो next codon है वो यहाँ पे होगा A site पे, और इसके response में आएगा tRNA next. So next amino acyl tRNA will bind at this site accordingly. So next tRNA will come to this A site. If we talk about translation in case of eukaryotes, it is very complex and it requires several soluble initiation factors. So eukaryotes के केस में translation का process complicated है और इसमें बहुत सारे soluble initiation factors required होते हैं. और जो first difference है, in eukaryotes amino group of the methionine is not formylated. So जो eukaryotes में जो first amino acid लाता है tRNA वो formylated नहीं होता है, as we have seen in case of prokaryotes. And initiation complex in eukaryotes is formed at 5' terminus of the mRNA. So there are no sequences like shine-Dalgarno sequences which are present in the eukaryotic mRNA. So the initiation complex moves through the mRNA starting at the 5' end, searching for an AUG initiation codon. So इसमें directly जो इनिशिएशन कॉम्प्लेक्स है वो मूव करता है mRNA के ऊपर, और जहाँ पे उसे इनिशिएशन कोडोन मिलता है वहाँ से वो स्टार्ट करता है फॉर्मेशन ऑफ अ पेप्टाइड. So eukaryotes have a special initiated tRNA that is abbreviated as tRNA small I. Here I stands for initiator. There is some special proteins which are known as cap binding proteins, which binds to the 7 methyl guanosine cap at the 5' end of the mRNA. And finally, the smaller subunit, that is 40th subunit, join the complex, and the entire initiation complex moves from 5' to 3' along the mRNA molecule searching for an initiation codon. So initially, initiation के time पे ये चीज़ होती है, and after that these enzymes move through this mRNA. When an AUG, that is initiation triplet, is found, the initiation factor dissociates. जो initiation factors है, जैसा हमने prokaryotes के केस में पढ़ा था, वैसे ये dissociate हो जाते हैं. And the largest subunit, that is 60th subunit, that binds to the tRNA, that is methylated tRNA, and mRNA 40th subunit complex, forming the complete 80th functional ribosome, जो कि protein synthesis में help करेगा in case of eukaryotes. Then next step is chain elongation, which is the second step in translation process. It is almost similar in both prokaryotes and eukaryotes. It occurs in 3 steps. First is binding of the specified amino acyl tRNA to the A site of the ribosome. So जो amino acyl tRNA है, जिसे charged tRNA भी कहते हैं, वो bind करेगा A site पे ribosome के ऊपर. Then second step is shifting of this nascent polypeptide. जो polypeptide है, that will shift from tRNA in the P site to the tRNA in a A site by the formation of a new peptide bond. So जो पेप्टाइड है वो शिफ्ट हो जाएगा P साइट से A साइट में, और A साइट के अमीनो एसिड के साथ बॉन्ड बनाएगा, which bond, that bond is known as peptide bond. So this shifting of this polypeptide chain occurs from the P site to A site. And third step is movement of ribosome along the mRNA to put the next mRNA codon on A site. So ये जो राइबोसोम का पूरा फंक्शनल राइबोसोम है, ये मूव करेगा mRNA के ऊपर इन द 5' to 3' डायरेक्शन, जिससे जो नेक्स्ट कोडोन्स हैं mRNA के, दे विल कम टू द A site of ribosome. So we will discuss these steps in detail. First step is binding of an incoming amino acyl tRNA. So this is the first step. जो amino acylated tRNA है वो bind करेगा सबसे पहले. So at the onset of elongation, जैसे elongation start होती है, and amino acyl tRNA as specified by mRNA binds to a A site of the ribosome. So जो mRNA के ऊपर कोडोन्स हैं उसके हिसाब से जो tRNA है वो bind करता है A site पे. This binding requires elongation factor. इसके लिए एक elongation factor चाहिए होता है, that is called TU, carrying a molecule of GTP. For this, GTP is also required. Then second amino acyl tRNA along with this elongation factor TU and GTP binds with initiation complex. This GTP is hydrolyzed and this GDP elongation factor TU is released and recycled. Then second step is peptide bond formation. जो second step है, here peptide bond is formed between two amino acid present at A and P site. So जो P site वाला अमीनो एसिड है ये शिफ्ट हो जाता है टूवर्ड्स द अमीनो एसिड ऑफ ए साइट, और इसके बीच में बॉन्ड बनता है जिसे बोलते हैं पेप्टाइड बॉन्ड. एंड दिस बॉन्ड इज फाउंड बाय एन एंजाइम व्हिच इज नोन एज पेप्टाइडाइल ट्रांसफेरेज. दिस एंजाइम हेल्प्स इन formation of this peptide bond, and this catalyzes this reaction of peptide bond formation found associated with the largest subunit, that is 50th subunit of the ribosome. और ये जो एंजाइम है ये एसोसिएटेड होता है लार्जेस्ट सबयूनिट के साथ. Then the third step is translocation. Here, ribosomes moves one codon towards 3' end of the mRNA, and this movement allows translation of dipeptidyl tRNA and deacetylated form from A to P and P to E. So जो ये movement है राइबोसोम की 5' to 3' डायरेक्शन में, इससे जो tRNA प्रेजेंट था A साइट पे, that will get shifted to the P site, एंड जो P साइट में प्रेजेंट है वो शिफ्ट हो जाता है E साइट में, दैट इज एग्जिट साइट, विच इज डीएसिटाइलेटेड. और वो जो tRNA होता है this is deacetylated. So this is this process of shifting occur or translocation occur, which is the third step in chain elongation. So movement of ribosome along with mRNA needs energy, which is provided by GTP and elongation factor G. This movement of peptidyl tRNA from A site to P site leaves A site. इस मूवमेंट की वजह से जो A साइट है ये पेप्टाइडाइल ट्रांसफर हो जाता है tRNA टू द P साइट, जिससे जो A साइट है दैट रिमेन्स एम्प्टी फॉर द नेक्स्ट tRNA molecule, thus allowing the ribosome platform to take up new charged tRNA, जिसकी वजह से जो नया charged tRNA that will enter into the A site and start another cycle of elongation. So this completes an elongation cycle. It occurs repeatedly until the stop codon arrives. और ये जो elongation का प्रोसेस है कंटिन्यूस चलता रहता है जब तक कि यहाँ पे टर्मिनेशन कोडोन ना आ जाए mRNA के ऊपर. The last step in case of translation process is chain termination, and this process is this termination is signaled by one of the three codons which are present on mRNA, which are UAA, UAG, and UGA, known as anticodons or termination codons or nonsense codons. So these three stop codons are recognized by soluble proteins. Special soluble proteins recognizes these stop codons, which are known as release factors, और इन्हें कहा जाता है release factors. So there are RF1 release factor which recognizes termination codon UAA and UAG. Then we have release factor RF2 which recognizes UAA and UGA. So these two release factors in case of prokaryotes help in termination, identifying termination codons on mRNA. In case of eukaryotes, there is only one release factor. यूकैरियोट्स के केस में सिर्फ एक ही रिलीज फैक्टर प्रेजेंट होता है, वो सभी तरह के टर्मिनेशन कोडोन्स विच आर थ्री टर्मिनेशन कोडोन्स उन तीनों को आइडेंटिफाई करता है. सो द प्रेजेंस ऑफ अ रिलीज फैक्टर इन ए साइट अल्टर्स द एक्टिविटी ऑफ पेप्टाइडाइल ट्रांसफेरेज. जो ए साइट है उसमें यहाँ पे रिलीज फैक्टर आ जाएगा इन रिस्पॉन्स टू एंटीकोडोन्स ऑन mRNA, जिससे जो एक्टिविटी है एंजाइम्स की दैट इज पेप्टाइडाइल ट्रांसफेरेज की एक्टिविटी अल्टर हो जाती है, means it can't forms the bond properly. So the enzyme then adds a water molecule to carboxylic terminus of the nascent polypeptide. और ये जो एंजाइम है वाटर मॉलिक्यूल कैरी करता है जो अमाइनो एसिड है, लास्ट अमाइनो एसिड उसके कार्बोक्सिल टर्मिनस पे इन द नेसेंट पॉलीपेप्टाइड जो पॉलीपेप्टाइड चेन बनी है. एंड दिस लीड्स टू रिलीज ऑफ पॉलीपेप्टाइड फ्रॉम द RNA मॉलिक्यूल इन द P साइट, जिसके रिजल्ट से जो P साइट से जो पॉलीपेप्टाइड है वो रिलीज हो जाता है बाय द एडिशन ऑफ दिस वाटर मॉलिक्यूल एट द कार्बोक्सिलिक टर्मिनस बाय दिस एंजाइम. सो इट रिलीजेस दिस पॉलीपेप्टाइड फ्रॉम द P साइट ऑफ राइबोसोम. Chain termination should also be understood to be an equally important step in protein synthesis as any undesirable addition to the polypeptide will also leads to production of abnormal protein. So this is very sensitive process. जो termination process है वो बहुत sensitive process है. उससे पहले इसको proof read किया जाता है. If there is any addition of abnormal amino acid that can results in production of abnormal protein. So this was all about this translation process. Hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.